Good morning, Professor. Tell us a little bit about your institution, your background, and the general scope of work that you're conducting.
Good morning. My name is Dr. Jack Sullivan. I'm with the University at Buffalo and the VA Medical Center in Buffalo. I'm a clinician scientist. My clinical interests are inherited retinal degenerations and macular degenerations. So I see patients who have these diseases both at the university and at the VA. In the research side, my lab is working to develop gene therapies for orphan retinal degenerative conditions as well as, for example, dry AMD. We are funded by the NIH, National Eye Institute. We're funded by the VA, currently by the State University of New York as well.
Tell us what's new for ARVO 2017.
We have 4 presentations at ARVO this year, and they all deal with optimizing a particular ribozyme type therapeutic that can be used for autosomal dominant retinitis pigmentosa due to mutations in that gene. But we also think that we can extend that same therapeutic into other disease states such as dry AMD and perhaps Stargardt.
What are the results that you'll be presenting here at this conference?
The results that we'll be presenting at ARVO 2017 are optimization trials to take the ribozyme, which is effective at cleaving the messenger RNA target. We have now put that ribozyme, which cleaves the RNA in vitro and suppresses the RNA target in cells. We have put that into a different RNA scaffold, which is how the ribozyme is expressed. And that new scaffold is a human small RNA scaffold, unlike the prior viral scaffold, which we found was toxic. So we're optimizing the ribozyme in that new scaffold, and we've brought in an array of collaborators and technologies, including structural biology and biophysics and molecular dynamics, to understand why does a particular ribozyme have the capacity to cleave its target once it binds, and probably more importantly, in the ones that don't work as well that we want to improve, how is that structure locally different from what we expect it to be from crystal structures, and how can we improve that structure of the RNA at an atomic level to make it a better enzyme. And so our presentations here, 2 of them are about that particular challenge as we move forward with it. In order for any kind of RNA therapeutic to be functional, it has to go to the same place in the cell that the target RNA is going. We call that co-localization. In fact, ribozymes can be an incredible knockdown agent if you specifically co-localize them with targets. There are studies in the literature where people have effectively achieved a knockout condition with hammerhead ribozymes. But you have to get them to the same place in sufficient concentrations. So we're developing methods in order to look at where the target mRNA goes in the cell, and we're able to label the target RNA with certain novel technologies. And by doing that, we can add fluorescent probes to the cell and see specifically where is the RNA showing up. And once we have that developed for the target, we're going to use similar approaches for the therapeutic to make sure that the therapeutic and the target are going to the same neighborhoods or zip codes in the cell where they can co-localize to further enhance the efficiency of the knockdown. And then finally, we have a bioinformatics paper that looks at where our current ribozyme and shRNAs are targeting in the human rhodopsin mRNA. And we've asked the question, does it matter in your experiments whether you're using a human mRNA rather than a dog RNA or a pig RNA or a mouse or rat RNA? And what we find is that the accessibility around our target site changes significantly when you use different mammalian mRNAs. So we are very concerned about taking a particular therapeutic that might have been shown successful in an alternative mammal and saying, oh, that's equivalently effective in expectation to move it into a human clinical trial. So we have a poster on Wednesday about that.
Returning to the scaffold, this is a scaffold for an in vitro model, or this is a scaffold that's intended for implantation?
The scaffold is another RNA. Right?
Okay.
It's a small RNA. It's actually an RNA that is normally made in abundance inside all human cells. And so what we do is we've engineered that RNA, which is a human RNA, and we've created an environment where we can put the ribozyme within that RNA, where it has flexibility and where the ribozyme could bind to the target within that scaffold and then undergo structural changes to align the nucleotide components to be able to do the cleavage of the target RNA that binds to it.
So how do you see this being delivered as a therapeutic option?
We are doing gene therapy. So this is not synthetic RNA chemistry. We are building genes that express these particular therapeutic RNAs with just standard 4-nucleotide chemistry of evolved biology. So we are using AAV-type genetic vectors, and our intent is to, our current protocols are using subretinal injections of AAV in a dry AMD or Stargardt model, and we're also using subretinal injections of AAV in an autosomal dominant, fully humanized mouse model that has a slowly emerging pigmentary retinal degeneration, and where the target RNAs are actually the same RNAs that would exist in a human clinical trial. We are developing ribozymes against the human rhodopsin mRNA. We're also developing ribozymes and shRNAs against other inherited retinal disease targets. And then once we have those ribozymes and shRNAs optimized, we will package them in AAV for subretinal injections or intravitreal injections once that technology gets to a point where photoreceptors or RPE can be very efficiently transduced.
Okay.
Since this is macular news, let me give you a little more about the rationale for what we're doing for dry AMD. Okay? Dry AMD emerges in the parafovea. This is a region of the retina where the rod density is starting to increase and the cone density is coming down. Okay? Any single RPE cell interacts with between 20 and 50 photoreceptors. So it has a huge metabolic burden to support those photoreceptors to process disc material and lipid retinoid byproducts on a daily basis. So our strategy is to say, well, we know that you can, in mouse models or even from human genetic studies, that you can reduce the amount of rod rhodopsin, not eliminate it, but reduce the amount of rod rhodopsin, even in patients, we expect. And by doing so, you would decrease the metabolic burden that is presented to the RPE cells. So by reducing the metabolic burden to the RPE cells, that can increase their vitality to support the overlying photoreceptors, and perhaps even increase the vitality of the cones because the 11-cis retinal will be more available to regenerate cone visual pigments. And then to the extent in dry AMD that the bis retinoid and toxic retinoid byproducts are a component that drives the disease, one would expect that reducing rod rhodopsin would also decrease the amount of those toxic byproducts. So that's the rationale. And so we're using a therapeutic that is developed for an orphan disease and translating that over to the identical target, but in the context of dry AMD, or perhaps as a supplement to treat autosomal recessive Stargardts, where the ABCA4 would need to be replaced, but it may be beneficial to reduce rod rhodopsin as well.
Now, as you know, AMD is a polygenetic disease, it's mosaiform. When you're looking at a repertoire of genetic interventions, what specific targets are you looking at?
That is true that there are a host of genes that create risk for people to develop dry AMD or wet AMD. Our approach is sort of independent of the genetics. So when you look at what are the stressors that create an environment in which dry AMD can emerge, if you change the nature of that system, right, and you change the rate at which those cells get sick, especially in a prolonged age-related, age-emerging disease, regardless of what the genetic background may be, if you relieve stresses, you can keep those retinal pigment epithelial cells and overlying photoreceptors likely alive longer into the life of the patient. Whether there would be any effects of various genes that have known AMD type risk, we don't know the answer to that. I mean, that's the kind of question that looks pretty far down the line. I think now, at the point where we're at, we know that there are variants that increase risk of AMD, but also there are a lot of genes that can contribute to risk. And just because you have disease-causing variants doesn't mean that you're going to develop AMD as well. So this is a long temporal scale in which dry AMD emerges, and we really only have the sense of the big brushstrokes of that at this time. So because of that, we have not gone after specific genes in a gene type therapy. We've gone to how do you change the system biology of the critical region of the retina where dry AMD and wet AMD emerges, to try to prolong the kinetics of the disease process. And if you have AMD increases over the age of 55, and so if you can change the kinetics and slow that down by 50%, for many patients that could ultimately be an effective treatment. So that's our strategy.
Yeah, that's a very elegant approach.
Thank you.
So going back to where you see the timeline for this particular research going, what are your endpoints for a year and from 5 years from now?
For the autosomal dominant RP, we are testing our optimized and enhanced ribozymes and shRNAs in a humanized model. We're actively doing that now. We're looking to move toward a successful optimization and an IND within the next perhaps 2 to 3 years. For dry AMD, we're beginning to test in an animal model that is really a Stargardt model. That particular model, we've worked out the conditions in which we can get a practical retinal degeneration on an experimental timescale, because otherwise it takes like 9 months to get a little bit of degeneration. So we use light to accelerate the degeneration. So we had to figure out how to use the light to accelerate the degeneration experimentally. And with the same approach, subretinal or intravitreal AAV delivery of the therapeutic ribozyme or shRNA genes, and then using electroretinography or OCT and histology and molecular approaches to assay where we're at. So there I think we're probably about 5 years out.
You've got a very elegant ecosystem that you're building around this approach, and I think that solidifies its strength.
Thank you. Well, I mean, the science doesn't happen in a vacuum, okay? I've been able and fortunate to be funded by different groups, and I have a very, very strong team with multiple people who are committed to this problem, and they're really the people that are, carrying the shoulders of this work. And so without the team, you don't get anywhere. So thank you.
Thank you, Professor. I really appreciate it.
Thank you very much, David.
Excellent.
Yeah.